Greetings to all the viewers. Now we are going to conduct the session based on the new concepts called the textbook of organic named reactions. This is the textbook written by, authored by Dr. Kavitha Kottireddy Kota. Okay, from the textbook we are going to discuss about various named reactions, what kind of reaction taking place, what significant applications it possesses, and what kind of possibilities of what changing that reaction. All these are taken into the consideration. This is the inception. So this is a starting reaction from which we are going to conduct the series of sessions based on textbook of organic named reactions, right? The entire content series of sessions available at the channel called World of Competitive Chemistry. Whatever kind of chemistry big points or is chemistry concepts you want to retrieve, all are available at the channel called World of Competitive Chemistry. Let me continue with this reaction called Chrysta-Venkata-Kavitha-Shrikrishna ring fusion reaction of coumarin compounds and their bioefficacy, right? So this is the kind of named reaction, Chrysta-Venkata-Kavitha Sri Krishna ring fusion reaction we are going to discuss in detail. Here, the author is Kavitha Kottireddy Kota. Then potential in the cytotoxic efficacy, hence essential to access and explore. So this is about the kind of introduction regarding the named reaction, right? Owing to the unconfined floor of extendability of coumarin research. Coumarin research finds multiple applications, multiple kind of resources available based on the coumarin research, being it is very mild and very potential in terms of medicinal applications, and moreover material chemistry applications also applied for this coumarin research. Distinctively applications of pharmacopoeia, advanced materials, even in the agricultural field you can apply coumarin related compounds, and in the industrial research this coumarin taken as a research objective, right? Desperate malignant tumor diseases are the global population threat it will be, right? So the tumor or else cancer will be the kind of distinctive factor which is adversely impacting multiple populations all across the global region, right? Universal and Indian health safeguard perspective, right? So not only in the worldwide context, you may consider Indian health safeguard perspective. It is very essential to improve bioefficacy profile to eradicate hepatocellular carcinoma. And in order to avoid this hepatocellular carcinoma, we developed a kind of Givold reaction. Coumarin thiazole furan nitriles were developed. Coumarin thiazole fused pyridine compound is also developed. So this is purely related with the eradication of the cancer tumor cells, right? Advanced screening studies were also conducted after synthesizing these compounds in the laboratory in the successful manner, in the environmentally viable manner. So they are performed for molecular modeling studies, molecular docking conducted based on BCL2 saline, and further confirmation of that bioefficacy against hepatocellular carcinoma is performed by IC50 examination. Here the saline used as a standard will be HepG2. HepG2 saline. Cancer saline was taken and it is treated with the various synthesized coumarin derivatives, whatever coumarin thiazole fused pyridine nitriles, whichever synthesized, performed against these cancer salines and successfully achieved the satisfactory results. So we performed the ring fusion reaction. This is a named reaction, and moreover, it is exhibiting potential application towards the cancer therapy, cancer treatment. That is the reason why this article is a continuation of the previous work and unique in the ring fusion reaction. Hence, being it is unique in nature, it is named as Chrysta-Venkata-Kavitha-Shrikrishna ring fusion reaction. Hence, undeniably, shrine of research track is suitable for further exploration and leads to appreciable pharmacological future curatives. Being it is highly potential towards the curation of the cancer, especially liver cancer. That is the reason why it becomes a lead compound, and you can extend the research, and further research analogs will be developed based on this named reaction, so that it is of high consideration, right? Here, graphical extract is provided. Coumarin is taken as a core pharmacophore, where you can notice the benzene fused with the pyran ring with the double bond O. This is the kind of cyclic lactone it will be. Liver cancer is the common feature. You can notice uncontrolled cell division leads to the tumor. Cell development, that kind of tumor is developed in the liver, so that will give the kind of acute health damage to the person who are suffering with this one. That is the reason why we gone through the successful establishment of this named reaction called Chrysta-Venkata-Kavitha-Shrikrishna ring fusion reaction, and the compounds whichever synthesized. So the various series of compounds got developed within the laboratory, and further we gone through their molecular modeling studies. Computer screening was conducted in order to understand the protein binding efficacy with that cancer cell, and successfully we achieved. There is a high what high strength towards that cancer saline binding tendency. That is the reason why we gone through the statistical studies as well, and finally achieved this compound whichever we synthesized, that is novel in nature, and moreover having high potency towards anti-hepatocellular carcinoma. The compound whichever synthesized is manifested over here. This is the one we successfully achieved for the synthesis in the laboratory, and subsequently treatment against the hepatocellular carcinoma. So this is the one which able to give the entire portfolio regarding this synthesis of the compound and subsequent application as well, right? As per the IUPAC nomenclature, the compound whichever synthesized is manifested as 5 amino, 7 furon, 2 aryl, 3 2 oxo, 2 H chromin, 3 I, 7 H thiazolo, 3 2 A pyridine, 6 8 dicarbonitrile is the IUPAC nomenclature accorded for the compound whichever synthesized in the laboratory by this named reaction, right? So this is about the introduction towards the cancer and also coumarin as well, right? So why was coumarin chosen for the cancer curation? So this is the most important segment regarding this inception of the new named reaction within the research of organic chemistry. Crunch of literature available across the globe. Every day new compounds will be added to the coumarin chemistry and expanding its profile, being it is highly flexible to explore, right? It is obvious that potential area of research and destination for the medicinal therapeutics at the global floor. Research exploration is most convenient. Not only that, whatever compounds synthesized are potentially in the medicinal application. That is the reason why there is a direct linkage between the new compound establishment and their further application in the field of medicinal and pharmacology, right? That is the reason why coumarin is very crucial in the research point of view and also the medication in disease curation, right? Proven potential pharmacopoeia profile is accorded for the compound. Evidence-based analysis and research, it is most authentic. Coumarin-based research is most authentic, and analysis is also a kind of simpler one. Simplicity in the synthetic routes and materials. It's a very convenient to synthesize within the laboratory, though we are not having sophisticated machinery, sophisticated environment, and a kind of establishment infrastructure may not be available. By using simpler routes at room temperature, at magnetic stirring, or else you may go for the water circulation, condensers, such mantle heating. By using very simple reactions, it is possible to achieve the new products based on coumarin compounds, right? On par high proficiency in therapeutic cancer treatment concerning the standard drugs whichever already available at the market. So that kind of efficacy they are exhibiting, whichever synthesized newly within the laboratory, those are based on the coumarin profiles, coumarin compounds, right? Provision for the structural modification and the transformation. Coumarin compounds are very viable, very easy ease of transformation is high for these coumarin compounds, so that multiple functionalities can be amended, can be established upon this coumarin, so that you can explore, you can improvise the structure of this coumarin, and you can connect with multiple functionalities, so that so many analogs will be newly introduced, and they may go for the therapeutic index. Being it is a phytochemical, negligible side effects. So you can extract the coumarin from the plant origin. That's the reason why side effects are very minimal. And human consumption is very safe for the consumption process, and being it is a mild compound. Right? Being it is a track of chosen research, prepared a library of coumarin-based compounds are exhibiting the successful strand of hepatocellular carcinoma. So that's the reason why this research is of high consideration, right? So coumarin compounds are having multiple applications in the pharmacological field, like they are having a kind of anti-cancer activity exhibited, anti-hepatic cancer in specific, anti-Lawrence cancer will be there, prostate cancer activity is also better curated, ovarian cancer, liver cancer, leukemia cancer, anti-sarcoma cancer activity, anti-tubercular activity, anti-Alzheimer's kind of brain-related one, anti-diabetic related with the insulin secretion and working of pancreatic gland, and antibacterial, microbial activity is also the proven activity shown by this coumarin. Antioxidant will be there, which used to boost the immune system, and melanogenesis. This is related with the skin irritation, skin cancer as well. So these many activities successfully established for this coumarin. So many compounds already introduced into the market to treat these many kind of diseases, and all are manifested over there, right? So this is the way coumarin plays a vital role in the field of pharmacology, right? So now we are going to synthesize the kind of new novel compounds based on Chrysta-Venkata-Kavitha-Shrikrishna ring fusion reaction. In continuation to the previous research, already much more research concerned regarding this coumarin thiazole hybrid compounds. We already performed within the laboratory, and much information got gathered, right? After that kind of reactivity, we are going to improvise the reaction. That's the reason why, continuation to previous work, conformed synthetic analogs were considered. Therapeutic investigations in the disparate pathways, various mechanisms got established. If the single compound synthesized in one route, so is there any possibility to synthesize in any other, all possible mechanisms in concern with the environmentally viable pathways, environmentally safe pathways? You may go for the room temperature, you may go for the solid phase mixing, you may go for the microwave, you may go for the ultrasound ultrasonication process or water mediated reaction. These many kind of possibilities, we gone through all these perspective in terms of that environmentally safe conditions in order to achieve the final product, right? So one method already successfully achieved, but optimization studies in order to get the effective way of synthesizing that compound is also very essential. Initially, in order to achieve the coumarin thiazole fused pyridine heterocyclic hybrid, multiple methodologies were designed. Not only single path, different schemes, same compound we can achieve in multiple ways. That those many methodologies got introduced. They are drawn, right? And not only drawing of those, they designed and finally executed as well. So, is there possibility to go for one by one stepwise mechanism for the synthesis? And also in an alternative pathway, same compounds mixed in a single reaction vessel tandemly, and we can achieve the compound within the single step. Multi-component single step process is also a kind of mechanism we can consider for the establishment of this novel new compound, right? This is the way. Multiple methodologies like stepwise traditional methodology, and also one component multi sorry uh, one step multi-component methodology is also introduced in order to achieve the compound in the short time with the less number of reagents. with a less number of steps, less number of intermediates without the formation of any other byproducts. This is the way purely considered in green reaction pathway. One pod three component methodology is best suitable. So that is indicated over here. If you want to observe, this is the key intermediate which is taken as a precursor for the synthesis of this novel compound. You may consider there is a coumarin moiety that is directly linked up with the thiazole 5-membered ring with the heteroatoms like sulfur and nitrogen at 1, 3 positions. And you can notice CH2CN that methylene nitrile is taken as a side chain upon the molecule which is simply said to be acetonitrile, where you can find this active methylene CH2 which is directly linked with the electron withdrawing functional group is said to be what highly reactive, more prone for the release of H positive. That is the reason why this is the key precursor for the synthesis of the target molecule. And melanonitrile is another active methylene compound where the methylene is directly connected with two nitriles. So two nitriles, if connected to the CH2, that is very viable for the release of H positive. So here the carbene establishment and here the condensation with a high rate of reaction, and another reactant whichever required will be aromatic aldehydes. Aromatic aldehydes are taken in the various forms. A to J kind of aromatic aldehydes to be taken, right? Simply you can go with the benzene ring that is said to be benzaldehyde. Not only benzaldehyde, but also chloro substituted, para-chloro substituted benzaldehyde may taken, para-bromo substituted benzaldehyde may taken, pyryl. Instead of benzene ring, you may apply heteryl aldehydes as well. That is the reason why there is a provision for the introduction of heterocyclic ring system over there. Pyryl aldehyde of uranyl aldehyde will be there, or else parahydroxy benzene phenyl can be taken, parahydroxy benzaldehyde may taken, paranitrobenzaldehyde may taken, thiophene instead of pyrrole ring and furan ring, there is a provision for thiophene as well, or else pyridine can be established over there. This is the way various aromatic rings to be introduced. That's the reason why not only the single product, rather you can achieve library of compounds where you can notice coumarin. Here is the thiophene with the 5-membered ring directly fused with the 6-membered ring with the nitrogen, which is said to be dihydropyridine, right? This is a fusion reaction where you can notice the novelty, right? Being the precursor compound is the novel, we only established this compound. That's the reason why whatever path we are following, whatever final product we are achieving, entirely said to be novel in nature. In order to achieve this one pot three component reaction, we introduced a solvent purely environmentally benign called ethanol and a triethylamine taken as a mild basic medium in order to establish the kind of ring fusion reaction. And reflex temperature is maintained 3 to 5 years reaction to be proceeded in order to achieve the final product. This is successfully achieved, and moreover the quality and purity of the product is extremely larger, so that this is the kind of reaction we are going through and final product is generated. Not only a single product, rather you can achieve 1 2 3 4 5 6 7 8 9 10 analog compounds are synthesized within the same pathway, within the same context of the reaction conditions, right? So this is the way one pot three component reaction in order to establish Chrysta-Venkata-Kavitha-Shrikrishna ring fusion reaction. So that scheme is provided for the better understanding. Later on, in another track of research, coumarin thiazole acetonitrile, this is a precursor you already aware about, that to be added with melanonitrile. And in addition, you may go for the reaction with benzaldehyde. These three are known in the earlier scheme. Along with these, you can add another additional component called sulfur powder. Sulfur powder is also mixed in the same reaction vessel. This is said to be one pot, four component reaction. And moreover, it is designated as a Gewald reaction. Gewald reaction where the establishment of thiophene ring is possible. in the presence of this kind of melanonitrile. And here is the kind of molecule called molecular sulfur and aromatic aldehyde. And here is the requirement is neither methylene. So this is the way another four component reaction taking place under same reaction conditions here. A certain distinct compound, a slight variation you can notice. Earlier you achieved the fused pyridine ring. Here the establishment of thiophene molecule is possible. Here is the coumarin. Here is the thiazole. Here is the thiophene ring is established by means of this Gavold reaction. Here also you are supposed to take various kind of aromatic aldehyde instead of benzaldehyde. In addition to the benzaldehyde, you may go for the substituted benzaldehydes and heteryl aldehydes as well. So that series of compounds again synthesized and a low compounds got established within the laboratory by means of same reaction condition. These many schemes you are supposed to perform within the laboratory. Later on, after synthesizing these compounds by successful pathways, environmentally viable pathways, you are supposed to go for. After synthesizing the compound, we need to go for how can we apply this compound? What is the use of this actually? Without using that compound, whatever research we are performing go waste only, because anything, anyone, any person should have certain extent of their own significance, their own importance, right? So that's the reason why whatever compounds we synthesize to be screened for molecular docking, molecular modeling studies got introduced by computer analysis. Computer screening was carried. These many compounds 4A to J means a scheme number one where ring fusion reaction be named as a new one, and this is a Gavold reaction products of IA to J. All these to be taken and tested against B cell lymphoma 2. B cell lymphoma 2 abbreviated as BCL2. So this is taken as the cancer saline, and this protein is directly treated with these series of compounds, so that liver cancer activity studies were conducted by molecular docking. So whenever molecular docking studies were performed, hydrogen bonding tendency with the protein, that chemical compound established binds with this protein of cancer saline by hydrogen bonding. Series of compounds are manifested with the docking scores among all those. 4I compound, which is exhibiting 56.09 kilocalories per mole. High efficacy for bonding studies were established whenever compound is 4I. So not only 4I but also 4H4J is satisfactorily exhibiting the better docking scores, right? So if you go with the second scheme, that is Gavold reaction, in that series of compounds, 5C and 5H are satisfactory towards this kind of molecular docking studies towards this cancer saline, liver cancer saline. If you want to see the docking scores of individual compounds, whichever we synthesized, for a compound docking score is manifested 42.01, whichever compound we synthesized in the lab is indicated over there. The newly introduced compound, its a corresponding docking score is provided within the tabular form among all the important one 4I, right? So that we need to focus. 4I compound is given with this coumarin, and here is the thiazole. Here is the fused pyridine ring you can notice, and here is the furon. So furon connected with this one, furon connected one. That compound is of high efficiency 56.09. It is the exceptional docking score towards the liver cancer activity. Whenever this is the lead compound is converted into the medication, effective medication, so that will definitely cure the liver cancer, definitely cure the liver cancer. This is the efficacy studies you are screening about. All these compounds, whichever we synthesized in the lab, all are performed against liver cancer activity, but exceptionally, furan containing this pyrrole fused system will give the exceptional molecular docking score. This means that this is more effective towards the cancer curation, right? So this kind of drugs are to be explored further. So this is the way screening studies were performed. And in order to study these screening studies, we are considering Nexabar as the standard compound. Nexabar score is provided Nexavar score already, this is already that marketed drug only, which already established in the market, whose docking score is 46.53 kilocalories per mole. But exceptionally, our new compound, synthesized compound, is having effective score against this cancer saline. So that this is more potential towards the liver cancer curation. This way, the screening examination is studied, comparative study. With respect to established drug, we are going to perform our own studies, our own screening. This is the way performance of docking studies were given for library of compounds. Not only that, you can observe how the docking profile is provided, how the compound is binding with this cancer saline. That a score is provided for this 4I compound. 4I compound, you can notice 3D structure of that one. That 3D structure is provided, and whose binding efficacy with this protein, cancer protein, you can notice hydrogen binding efficacy is provided within this molecular docking profile. And this is for another Givold reaction containing compound. Later on, confirmation of anti-cancer activity. Initially, we gone through the identification, preliminary examination got performed. After getting the particular idea about the cancer curation, we need to go for the confirmation. How can we confirm? We performed IC50, IC50 studies were conducted by taking hepatocellular cancer, HepG2, hepatocarcinoma cell, HepG2 saline was taken. This is a standard one for the cancer, liver cancer affected saline was taken. And upon that, you're going to perform the cancer screening by introducing the newly synthesized compound. These many compounds we synthesized, right? Again, here that kind of IC50 score to be taken among all the compounds. IC50 values, if you are noticing, lower the IC50 value, higher the efficiency of compound. Here is the reciprocal studies you are going to examine. So that lower the IC50 value, higher will be the efficiency of the compound. 10, 12, 10, 13, 18, 11, 10, 9, 8. 9.8 is more potential. If you observe this one, 8.76. 8.76 again is the consideration for the better cancer curation. That is the reason why. So, for again taken into the consideration where the coumarin thiazole, fused pyridine ring, which is in contact with the pyrrole moiety. With a high IC50 score, you can notice again this kind of studies were conducted in comparison to the standard drug Nexavar, which is the anti-liver cancer activity drug. So this is the way we successfully accomplished preliminary studies and confirmatory studies for this anti-hepatocellular carcinoma by means of IC50 screening. So this is the way the entire tabular form is provided, and those results will be graphically represented. The statistical data is also provided. Various concentrations were taken and viability studies were conducted. There is a linearity, exponential decrease as we are increasing that concentration. You can notice clearly 4I is the important compound that is more potential, we already noticed. And hepatocellular G2 saline was taken as a standard on which we are operating the compound, so that here curation studies were studied, right? So here molecular docking is conducted by taking these series of synthesized compounds are the testing compounds, and BCL lymphoma is the kind of cancer saline you're considering. And molecular docking studies were performed within the computer screening platform. So the three-dimensional representation of BCL2 protein domain is taken, and that is treated with the novel compounds whichever synthesized in the laboratory. This is the BCL2 saline 3D profile. You can clearly notice upon which our drugs are operating, and active site identification is possible. So if this is the kind of protein structure where the active site, where the actual binding taking place, that kind of studies you can notice within this structure. So this is the way we performed anti-hepatocellular carcinoma studies. IC50 quantification got completed. So here Anti-hepatocellular carcinoma activity of these synthesized compounds were investigated, here G2 carcinoma cell lines, determined by measuring number of live cells after 24 hours of the treatment. MTT assay got conducted compared with the standard trucnexavar. This is the kind of activity we performed, successfully achieved. First step will be the synthesis of compound. That is one effort. We already got successful results over there. Later, the synthesized compounds are exhibiting potential activity in the liver cancer treatment. This is the way the reaction is very high concern for the further research exploration. This article, synthetic organic chemistry, part of synthetic organic chemistry, extended conjugation is provided within the molecule. That is a key factor for the higher efficacy towards the anti-cancer, anti-hepatocellular tumor activity is provided over there. Scientific and technical studies together performed over there. We also performed within the laboratory. Chemical identification got conducted in the scientific perspective, and the technical studies also gone through so many machineries. Analytical tools got utilized within the system in order to achieve the results in the satisfactory manner. Coumarin thiazole thiophan fused pyridine conjugates generated in the successful manner, and anti-liver cancer activity potency established, and quantified values you already noticed, and the confirmatory studies IC50 scoring is also taken part of that one, so that this is the way the entire research conducted, and we successfully established the new named reaction, Chrysta-Venkata-Kavitha-Shrikrishna ring fusion reaction of coumarin compounds and their bioefficacy. And bountiful of literature screening got conducted in order to achieve this kind of reaction. We thankful towards that kind of all the research material which already available, the kind of research which already conducted over there, and kind of authentic sources of journals, authentic sources of research platforms. We are very much sincerely thankful for all these kind of platforms. Thank you one and all for your patient listening.